Hi Nat5s, so we're looking at the structure of DNA as part of the cell biology unit. So DNA, I'm expecting you have heard of. It is this shape, and you see it all the time being used for all sorts of things. It stands for deoxyribonucleic acid. Here's your D, here's your N, and here's your A. Okay, but you're okay with just saying DNA. What you do need to recognize is its shape, and so this shape is called a double-stranded double helix, because what you have is two different strands. Obviously you can see this one running down this way, and the second one running also. Technically it's going up the way, but you don't need to know that till higher. Okay, so you've got two strands, and then they're twisted together to give this very specific shape, and it has things like it's got a narrow section here and then a wide section here and then another narrow section. That's part of the shape. So I think the simplest way to think of it is if you hold one end of a ladder and twisted the other end, this is the shape you would get. Every single rung of the ladder, if we go by that, so the bits going across the way here, is made up of two bits, one linking from one side and one from the other side, sticking together in the middle, and that's what's holding the whole thing as a double helix. Okay, and those are called bases, and you need to know those in a little bit more detail. Okay, so the bases, 4 types: adenine, which always pairs with thymine, and guanine, which always pairs with cytosine. You actually need to know the full name. You're not going to get away with, most of the time, you might get away with saying A, but a couple of years ago they asked you for the name of the base and you had to give adenine, you could not just say A. So generally we just say A to T and G to C, and that's fine, but just make sure you actually know the names of the bases as well, because they could be that precise. Okay, so you could be shown it lots and lots of different ways. This is a flattened out version, but it will always be A to T, like here, and G to C. Okay, so why do these bases actually matter? Why does it matter exactly what the code is? And it's because the code is coding for something in particular, and the code is the amino acid sequence. And the amino acid sequence decides protein structure, and proteins ultimately do the work. Okay, these are the ones in the cell that make things happen. So you need to know a reasonable list of things that proteins can do. Some examples are structural, like they hold the cell in a particular shape. Enzymes, we're going to do a lot more on these later. So they are catalyzing reactions, they're making reactions speed up, either ones that break things down or ones that build things up. Hormones, peptide hormones are a whole class of hormones, so communication within the cell and to other places. Antibodies are essential as well, so that's part of your immune system, and they can be receptors on the plasma membrane. I would also add in here, as well as receptors in the cell membrane, we had in the last subtopic carrier proteins, and we also had pumps that are being used for active transport. So both of these are also really good examples if they ask you what can proteins do inside a cell. So to be clear on how this is working, each section of DNA that codes for a particular protein is called a gene. Okay, and that gene is not going to be small, it's not going to be a few letters, we're talking thousands, so you normally actually talk about them in kb kilobases. Okay, so 1,000 bases long. And the forms of the gene you inherit determines what your cells can do. That should not surprise you. Okay, so which ones you get decide what you actually have in your chromosome, decides what you can then produce to make things happen. Okay, that's the overview. That's what you need to know.